All right, let's talk about something called non-aqueous titration. I know, it sounds a little intimidating, right? But stick with me because it's actually a super clever fix for a really common headache in chemistry. And believe it or not, this problem starts with the one thing we always think of as the solution: water. In chemistry, we're taught that water is the universal solvent. It's the hero, right? It mixes with everything. But what if it's not? What if for certain jobs, water is actually the villain? What if it's the very thing that's messing up our experiments, making it impossible to get a clear answer? That's the big question we're diving into. Okay, so let's get right to it. What's the problem with water? I mean, how can it possibly be a bad thing for chemists? So here's the deal. A lot of really important chemicals are what we call weak acids or bases. And when you try to dissolve them in water, well, they just don't play ball. They're stubborn. They refuse to fully break apart or dissociate, which is what they need to do for us to measure them properly. So when you try to do a standard titration, you never get that crystal clear endpoint you're looking for. It's like trying to spot a finish line in a super dense fog. It's just blurry, useless.
But, chemists are a clever bunch. They looked at this foggy mess and thought, well, if the water is causing the fog, why don't we just get rid of the water? It's a simple idea, but a brilliant one. Let's check out the solution.
And that brings us to non-aqueous titration. The name sounds fancy, but it literally just means titration without water. The whole idea is to swap out water for a different solvent. A solvent that those weak acids and bases actually like, one that lets them react the way they're supposed to.
Okay, so what does this actually look like in the lab? It's really straightforward. Step 1, you take your sample, that weak acid or base, and you dissolve it in a special solvent that isn't water. Step 2, you just do the titration like you normally would. But this time, boom, you get that sharp, obvious endpoint you've been looking for. The fog is gone. It's that simple. Now, this isn't a one-size-fits-all kind of deal. Think of it more like a toolkit. You've got two main tools, each designed for a specific job. Let's meet them.
Yep, there are two main methods, and which one you use is super simple to figure out. It all depends on if you're working with a weak acid or a weak base.
The two methods are called acidimetry and alkalimetry. It's easy to keep them straight. For weak bases, you use acidimetry. For weak acids, you use alkalimetry. Let's break down acidimetry first.
All right, so first up, we have acidimetry. This is the one you pull out when you're trying to measure those stubborn, weak bases that just won't behave in water. The goal is simple. Figure out exactly how much of a weak base you have. And you do that by titrating it with a strong acid. So it's still the classic acid meets base reaction. We've just changed the scenery to a non-water solvent where things can actually happen. And this is where it gets really important. We're not just talking about abstract lab experiments. This is how we accurately measure things like epiphedrine, morphine, adrenaline, even the caffeine in your morning cup of coffee. For stuff like that, you can't afford to be kinda right. You have to be precise. So, how do you see the endpoint? You need an indicator, something that changes color. A really common one for acidimetry is crystal violet, and it gives you this awesome, unmistakable color change from a deep violet to a light green. When you see that green, you know you're done. Perfect. Okay, so that's for bases. What about the other side of the coin? That would be alkalimetry. And you've probably guessed it, this is the method we use for weak acids. It's basically the mirror image of what we just talked about. Here, the goal is to measure a weak acid, so we do it by titrating with a strong base. Same principle, just flipped. And the applications here are huge, too. This is super important in the food industry. We're talking about checking the acidity of oils, milk, even wine. It's also used for measuring important medications like the gout treatment allopurinol. So, yeah, pretty vital stuff. And just like before, you need an indicator to see what's going on. For alkalimetry, a popular choice is thymol blue. Again, it gives you a nice, clear color change so you know exactly when to stop. Okay, let's do a quick recap to lock this in. Acidimetry is for weak bases, and you use an acid to measure them. Think measuring caffeine. Alkalimetry is for weak acids, and you use a base to measure them. Think checking the acid in milk. See? Two sides of the same coin. A really neat and logical system. So we've seen the problem, water creating that foggy mess, and the clever solution of just not using water. But let's take a step back. Why is this so important? What's the big picture here? This really says it all. The fact is, a huge number of things we depend on every single day, medicines that save lives, the food on our tables, are weak acids or bases. If we couldn't measure them accurately, things like quality control, safety testing, and scientific research would just stop. It's that fundamental. So, at the end of the day, non-aqueous titration isn't some dusty, forgotten technique. It's a critical tool. It's what stands between us and impure drugs. It's how we know our food is safe. It's all about making sure we can be absolutely precise when it really, really matters. And that's what I want to leave you with. The biggest lesson here isn't just about titration. It's about how a huge breakthrough can come from flipping a simple assumption on its head. In this case, realizing that water, the universal solution, was actually the problem. It really makes you think, doesn't it? What other basic rules are we following that might be holding us back? What else could we discover if we just dared to change the environment?